Whenever we try to, conceptualize a forest, I feel like we all suffer from this massive case of terrestrial bias.
Oh, absolutely. Yeah, it's a completely understandable visual heuristic, really.
Right. You walk into the woods, you look up at that huge, towering canopy of oaks or pines or spruces blocking out the sun, and the natural conclusion is just, well, the trees are the main characters.
Exactly. We equate sheer biomass above the ground with ecological control.
Yeah, we totally categorize the world by what we can physically bump into.
Right. But from a mycological perspective, that whole skyline is basically just the exhaust manifold.
The exhaust manifold, I love that.
Yeah, because the actual engine of the forest, like the biological infrastructure directing the flow of energy and minerals and water, that is entirely subterranean.
Which brings us to the mission of our deep dive today. We are stripping away that terrestrial bias for you, the listener, to look at a highly specific, honestly bizarre little architect of this underground engine.
And a really fascinating one at that.
Right. We are talking about the genus Hydnum, which most people just know as the hedgehog mushroom. Specifically, we're focusing on the true hedgehog, Hydnum repandum, and its close cousin, the terracotta hedgehog, Hydnum rufescens.
Yeah, and we should clarify right away, this isn't just some niche botanical profile we're doing here.
No, not at all. We've got this massive stack of recent multi-locus taxonomic surveys, ecological papers, and biochemical analyses powering this deep dive.
Including Tulinus Cannon's landmark 2018 study, which really kicked all this off.
Exactly. And the sources reveal an organism that is, and this sounds like sci-fi, actively dissolving solid rock. It's running this encrypted underground resource exchange, and it's even synthesizing complex neuroprotective chemicals that might have, profound implications for Alzheimer's disease.
Yeah, the chemistry alone is mind-bending.
Right. Plus, the crazy fact that for over 2.5 centuries, the greatest scientific minds completely misunderstood its global evolutionary history.
They really did. The sheer volume of data we have now has forced a complete paradigm shift. We're looking at this perfect intersection of mistaken historical identity, ancient DNA, plate tectonics, and, well, the brutal economics of forest survival.
So let's bring you into this detective story. And let's just start with the physical organism itself, because it immediately breaks the fundamental rules of what most people expect a mushroom to look like.
Right, it really defies expectations.
Like, the genus Hydnum was classified way back in 1753 by Carl Linnaeus.
The father of modern taxonomy himself.
Right, and he set Hydnum repandum as the type species. But if you pick one up in the woods, you don't find those classic gills you'd see on, say, a grocery store portobello.
No, not at all.
And you don't find the spongy pores of a porcini either. What you find is a spinose hymenophore.
Which is a very technical way of saying it has teeth. Foragers affectionately call it the sweet tooth of the forest.
The sweet tooth. Yeah, it's thousands of these downward-pointing, really brittle little spines just hanging off the underside of the cap.
Yeah, and the hymenophore, just for context, is simply the spore-bearing surface of the fungus.
Okay.
From an evolutionary engineering standpoint, the whole goal is to pack the maximum amount of surface area into the smallest, most protected space under the cap, right?
Right.
Because that's where you produce spores.
Right, more surface area equals more spores.
Exactly. Gills accomplish this with parallel plates. Pores use vertical tubes. But the Hydnum lineage, they evolved hanging stalactites.
Okay, let's unpack this for a second. Why evolve teeth? If gills and pores are overwhelmingly the dominant, successful architectures across the whole fungal kingdom, is a spinose structure just a less efficient evolutionary quirk, or is there some brilliant adaptation we don't fully get?
Well, it's actually an incredibly elegant solution to fluid dynamics.
Really? How so?
So, fungal spores have to escape the dead air space immediately under the cap. We call it the boundary layer. They have to get out of there to catch the microcurrents of wind that carry them away.
Okay, that makes sense.
Gills can sometimes trap spores, especially if the air is too still or if moisture causes the gills to swell up and get too close together.
Oh, interesting. So they get stuck.
Exactly. But the spines of the hedgehog mushroom utilize pure gravity. Because each tooth tapers to a microscopic point, the spores basically just detach and fall straight down.
With no structural obstruction around them.
Right. It is a highly efficient gravity assist mechanism, and it's completely distinct from the gill architecture.
Wow. And that structural distinction doesn't just stop at the teeth, right? It extends deep into the actual flesh of the mushroom itself. The sources classify the physical tissue of the hedgehog as monomitic.
Yeah, monomitic tissue. This is crucial. So fungal bodies are made up of these microscopic thread-like filaments called hyphae.
The basic building blocks.
Right. In a lot of tough woody mushrooms, you find a very complex matrix. You have generative hyphae, which are the main blocks, but then you also have thick-walled skeletal hyphae for structural support.
Like actual scaffolding.
Exactly. And then highly branched binding hyphae weaving it all together. That's a tremitic structure. It creates something nearly unbreakable, like a polypore that you see growing on a dead log.
But the hedgehog is monomitic, meaning it relies on just one of those threads.
Exactly. It consists entirely of generative hyphae. These are just thin-walled, inflated cells. Imagine stacking thousands of microscopic, elongated water balloons together.
Without any of the fibrous skeletal support.
Right, so the flesh of the mushroom is incredibly brittle. It has this almost chalky texture. If you apply lateral pressure, it doesn't bend or tear. It cleanly snaps.
Which is a major feature for foragers in the field, right? Because there are other mushrooms out there with teeth, generally like Sarcodon or Phelodon.
Right, the leathery tooth fungi.
But those have complex hyphal structures making them tough or corky, so if it doesn't snap perfectly like a piece of chalk, it's not a true hedgehog.
Exactly. But here's the thing. From a historical scientific perspective, this really simple monomitic structure created a massive headache for taxonomists.
Because they were just looking through early microscopes, right?
Yeah. It created literally a 200-year bottleneck in classification. When you look at those generative hyphae under high magnification, you see they utilize something called clamp connections.
Okay, let's visualize a clamp connection for the listener, because the sources describe this in a really cool way. Fungi have this fascinating reproductive state where a single cell contains two distinct unfused nuclei.
The eukaryotic state, yeah.
Right, and when that cell needs to divide and grow, it has to make sure that the new daughter cell gets exactly one copy of each nucleus. But doing that inside a microscopic tube, maneuvering two nuclei past each other, that's like a geometric nightmare.
It really is. So the fungus creates a literal physical bypass tube.
It's just so clever.
It's a brilliant adaptation. The hypha builds this microscopic backward curving branch, basically a peg that acts as a detour.
So one nucleus goes through the main tube.
And the other divides through the bypass peg. This ensures the correct distribution of genetic material right before the cell wall seals them off. It's like a microscopic ballet.
That's incredible.
We see these clamp connections abundantly in Hydnum, but what we do not see are cystidia.
Ah. And the lack of cystidia is what left scientists totally flying blind.
Completely blind.
Because cystidia are those large, specialized, sterile cells that stick out from the gills or pores of other mushrooms, right?
Yes, and they're wildly varied. Some act as structural spacers, some exude defensive chemicals. And because their shapes are so unique to specific species, taxonomists use them as microscopic fingerprint markers.
But without cystidia, the microscopic landscape of the hedgehog mushroom is just plain.
It's essentially a featureless plane. You just have a uniform sea of basidia, which are the cells generating the spores.
So stripped of any unique microscopic architecture, those 18th and 19th century mycologists were basically forced to rely almost entirely on physical appearance macromorphology to tell species apart.
Yeah, they had nothing else. They had to look at cap color, overall size, and how the spines attach to the stem.
Which perfectly brings us to the historical division between our two main subjects. The true hedgehog, Hydnum repandum, and the terracotta hedgehog, Hydnum rufescens.
The classic visual dichotomy.
Right. So the historical text defined Hydnum repandum as the larger, fleshier one, pale cream or light orange cap. But most importantly, its spines are decurrent.
Right, decurrent meaning they don't stop at the stem. They actually run down the stalk like a little waterfall.
Contrast that with Hydnum rufescens. It's much more slender, smaller. The cap is a deeper russet or terracotta color, and the spines are adnexed.
Meaning they stop abruptly at the exact point where the cap meets the stem.
Add in the fact that the spores of rufescens are fractionally larger and more broadly ellipsoid, and, well, that was it.
That was the entirety of the defining parameters.
Yeah.
For over 2 centuries, if a scientist literally anywhere on Earth found a large, pale hedgehog with decurrent spines, they logged it as Hydnum repandum.
And this right here is what entrenched the idea of a holarctic distribution.
The big myth.
This massive assumption that these specific fungal species were basically cosmopolitan globetrotters, just blanketing the entire northern hemisphere across North America, Europe, and Asia.
Right, based entirely on the fact that these mushrooms wore the same physical coats, which completely sets the stage for the taxonomic revolution.
Let's get into that revolution because it shatters everything we just talked about.
Prior to modern molecular phylogenetics, the global consensus recognized roughly 12 species of Hydnum total.
Just 12 for the whole planet.
Right. The paradigm was that fungi reproduce via microscopic spores capable of catching upper atmospheric currents. So the assumption was they could simply drift across oceans and colonize anywhere with a suitable climate.
But then the 2018 Niskanen study systematically dismantled that holarctic myth. And they didn't do it by just looking closer at the cap color, obviously. They used multi-locus phylogenetic analysis,
Which changed the game entirely.
Let's break down the mechanics of this for everyone listening, because it is vital to understand how we know the history of biological life has to be rewritten here. They didn't just use a single genetic barcode.
No, they sequenced multiple specific regions of the fungal DNA. They targeted the ITS region first, the internal transcribed spacer.
Which is the standard one, right?
Yes, it's the standard barcode for fungi because it sits between the ribosomal RNA genes and it mutates rapidly enough to distinguish between closely related species.
Okay, but ITS alone isn't enough to map deep evolutionary time.
Exactly. So they paired it with the NRLSU, which is the nuclear large subunit of ribosomal RNA, and highly specific protein-coding genes like translation elongation factor 1 alpha or TEF1 alpha.
Okay, TEF1 alpha. This is where I think we need to address some common skepticism, because when scientists rewrite 250 years of established biology basically overnight, it is fair to ask, how does sequencing a protein-coding gene like TEF1 alpha provide absolute mathematical proof of evolutionary divergence?
It's a very fair question.
Like, if two mushrooms look identical, they behave identically in the forest, they grow in the same climates, isn't it possible the DNA test is just splitting hairs?
Well, it's really the difference between observing convergent evolution and reading a molecular clock.
A molecular clock, explain that.
Visual traits, like having a pale cap or decurrent spines, those are heavily influenced by environmental pressures. So if the environment in Oregon and the environment in France both demand a specific physical adaptation for a mushroom to survive, well, those mushrooms will eventually look identical.
That's convergent evolution.
Right. But the genes we target in multi-locus sequencing, they act as an evolutionary diary, specifically because of how they accumulate mutations.
Because they utilize synonymous mutations. I remember reading this in the sources. The genetic code is redundant, right?
Yes, highly redundant.
So you can have a mutation in the DNA sequence of a gene like TEF1 alpha that doesn't actually change the final protein that the gene produces.
Exactly. And because it doesn't change the protein, it doesn't affect the mushroom's survival at all.
Which means natural selection doesn't read it out. It just sits there.
Precisely. These neutral, synonymous mutations accumulate at a remarkably steady, predictable thermodynamic rate over millions of years. It literally acts as a biological metronome.
Wow.
So by aligning the DNA sequences of a hedgehog mushroom from Oregon and one from France and counting the number of these specific genetic discrepancies, we aren't just nating differences.
You're calculating time.
We are calculating the exact geological epoch when their last common ancestor existed. When the data shows a massive sequence divergence in regions that normally only vary by fractions of a percent within an interbreeding species, it's irrefutable.
It's absolute proof of millions of years of genetic isolation. The numbers just don't lie.
They really don't.
So when the Niskanen team applied this multi-locus analysis globally, those original 12 recognized species just vanished and the numbers skyrocketed.
It exploded. The initial study instantly verified 49 distinct phylogenetic species.
49 from 12.
And it gets crazier. Subsequent environmental DNA sampling, where researchers sequence DNA directly from soil samples without even needing to find the physical mushroom, has pushed that number to 84.
84. That is a massive jump.
And statistical modeling suggests we haven't even cataloged half the global diversity yet.
Okay, so what was the bombshell regarding our classic European pair? The true hedgehog and the terracotta hedgehog.
The bombshell is that Hydnum repandum and Hydnum rufescens are strictly exclusively endemic to Europe.
They don't exist anywhere else.
They do not exist in North America. They do not exist in Asia, period.
So if you're listening to this in the U.S., every single field guide printed in the United States before 2018 telling foragers they were picking Hydnum repandum was fundamentally incorrect.
Completely wrong. The North American hedgehogs are entirely separate species that have been isolated from Europe for epochs.
That is just wild to me. It's like finding out the cousin you've been pen pals with in America isn't actually your cousin, but a genetic doppelganger who just happens to wear the exact same terracotta coat.
That's a perfect analogy. And the cryptic diversity in North America alone is just staggering. We are talking about 17 distinct phylogenetic species just in the eastern United States and Canada.
17.
Yeah, species now formally named Hydnum vagabundum, Hydnum subcanatum, Hydnum ferruginescens. And to the naked eye, they are practically indistinguishable from the European variants.
And what about the West Coast?
In the West, you have completely different endemic lineages, like Hydnum washingtonianum and the recently described Hydnum oregonense.
Okay, but when you look at the global picture, like Asia and Australasia, the genetic timeline gets even deeper, doesn't it?
Oh, much deeper. The study revealed that Asia, specifically China, is this massive hotspot of ancient diversification, entirely new clades like Hydnum bifurcatum. But Australasia, like Australia and New Zealand, holds what researchers literally termed strongly divergent old relics.
Old relics. So we're talking about living fossils of the fungal kingdom here.
Exactly. Lineages like Hydnum crossidens in New Zealand. The molecular clock data indicates these lineages have persisted in absolute genetic isolation since the tectonic breakup of the supercontinent Gondwana.
Tens of millions of years ago.
Right. They literally rode the tectonic plates to their current locations and never interbred again.
Okay, but the sources also show evidence of much more recent continental hopping too, right? Because the genetic trees show some lineages utilizing the Bering Land Bridge or those North Atlantic land bridges during periods of glaciation, just migrating back and forth between Asia and North America.
Yes, there's a mix. So you have a global map of ancient, deeply isolated Gondwanan relics sitting right alongside relatively recent intercontinental migrants.
It creates this beautiful, complex map of Earth's paleoclimatology, basically written entirely in the DNA of a forest mushroom.
That's incredible.
But this raises a glaring ecological paradox, and I think we really need to dig into this. We noted earlier that fungal spores are aerodynamic marvels.
Yes.
A single mushroom can drop millions of spores, which are light enough to be swept into the stratosphere and cross oceans. So if the dispersal mechanism is global, why is the genetic reality so violently localized?
That's the big question.
Right, like, why did the European species stay locked in Europe while the American ones speciated into 17 different localized versions? If the spores can literally go anywhere, why didn't they colonize everywhere?
This right here is the critical transition from taxonomy into ecology. The spores do, in fact, travel globally. That part is true. But a spore landing in a new environment is completely meaningless if it cannot survive. The Hydnum genus is biologically chained to the local flora. They are obligate ectomycorrhizal fungi.
Okay, obligate ectomycorrhizal. Let's break down the mechanics of that biological chain for everyone, because obligate means it is an absolute requirement for survival, right?
Yes, cannot live without it.
An ectomycorrhizal describes a very specific architectural relationship between the fungus and a host tree.
Exactly. See, most fungi people are familiar with, like oyster mushrooms or shiitakes. Those are saprotrophic.
Meaning they eat dead stuff.
Right, they possess the enzymes to break down and digest dead wood or organic matter. But Hydnum species completely lack those enzymes.
Really?
Yes. If a hedgehog spore lands in a pot of the richest, most fertile, sterile potting soil on Earth, it will germinate, send out a microscopic search thread of mycelium, and then rapidly starve to death.
Wow. Just dies.
It requires the chemical exudations of a living, compatible tree root to survive. It associates primarily with specific conifers like pine and spruce, or broadleaf trees like beech and oak.
It's basically born hungry for a partner, not for food.
That's a great way to put it.
Yeah.
And when that microscopic hyphal thread actually encounters a compatible fine root tip in the soil, the architecture they built together is astonishing, because it doesn't pierce the tree's cells like a parasite.
Right. The sources specify it forms an ectomycorrhizal sheath. The fungus heavily wraps the exterior of the root tip, creating this dense, protective glove called the mantle.
Yes, the mantle. And it acts as a biological customs checkpoint, regulating everything that enters and exits the root.
Okay.
From this mantle, the fungus pushes these microscopic threads inward, navigating the labyrinthine spaces between the cortical cells of the root. This massive, intricate exchange interface is called the Hartig net.
Named after Theodore Hartig, the 19th century botanist.
Exactly. And the Hartig net is where the subterranean economy actually operates. The tree has a monopoly on solar energy, obviously. It photosynthesizes, pulling carbon dioxide out of the atmosphere and synthesizing simple liquid sugars.
But the tree's massive root system is actually incredibly inefficient at scavenging for trace minerals, right? Especially the ones locked inside microscopic soil pores.
Yes, the roots are way too thick.
Yeah.
And they lack the chemical tools to extract minerals from solid inorganic matter. So the tree acts as a carbon battery. It pumps its synthesized sugars down into the Hartig net.
To pay the fungus.
Right. And the fungal mycelium, which is thousands of times finer than a tree root, acts as an external stomach and an industrial mining operation. It explores the soil volume, pulls in water and vital nutrients, and trades them across the Harding net for the tree's carbon.
But this isn't just passive scavenging, and here's where it gets really intense. The literature actually refers to Hidnum as an ecological engineer. They aren't just scavengers, they are deep earth miners and stockbrokers.
They are heavily engaged in chemical warfare, essentially.
Because the fungus isn't just soaking up loose minerals, it is literally dissolving the geology of the forest. Let's explain how oxalate and citrate actually melt solid stone, because that is wild.
It's fascinating chemistry. The fungal hyphae actively synthesize and exude powerful low molecular weight organic acids, predominantly oxalic acid and citric acid.
Okay.
When these acids encounter solid rock, we're talking minerals like feldspar or apatite, a process of chemical weathering called chelation occurs.
Chelation?
Yes. The negative charge of the oxalate molecules physically pulls positively charged cations, like calcium, iron, or magnesium, right out of the rock's mineral lattice.
It strips the structural integrity of the stone away at a molecular level. It literally causes the rock to degrade.
Exactly. And simultaneously, the fungus secretes specialized enzymes called phosphatases.
Because a massive amount of the phosphorus in a forest is locked up in dead, decaying organic matter, right? In these complex ester bonds that the tree just cannot utilize.
Right, the tree can't touch it. But the phosphatases chemically cleave those bonds, freeing the inorganic phosphate. The fungus then shuttles these heavily mined, newly soluble minerals through miles of its mycelial network.
Back to the Harding net.
Back to the Harding net and trades them to the tree.
It is a fully encrypted chemical exchange. The sources actually point out that the tree audits the fungal delivery. If the fungus stops providing phosphorus, the tree straight up restricts the carbon flow.
It's a brutal market economy down there.
And this hyper-specific mining operation perfectly answers our geographic paradox from earlier. The reason there are 84 distinctly localized species of hedgehog mushroom is niche specialization.
Precisely. Because the soil chemistry in the Pacific Northwest is fundamentally different from the calcareous soils of a French beech forest.
Right.
Over millions of years, the local Hidnum lineages evolved highly specific biochemical toolkits to dissolve the exact mineral profile of their local geology, while perfectly matching the chemical communication signals of the specific local trees.
So the newly discovered Hidnum organins isn't just randomly growing in Oregon. It has spent millions of years fine-tuning its organic acids to work in that specific soil, specifically trading with the bishop pine.
Exactly. If you take its spores and drop them in Europe, its chemical keys simply won't fit the local biological locks, and it dies.
Wow. It really shows how you can't just move these organisms around.
And this localized specialization is the invisible scaffolding of the entire ecosystem. The mycelial networks also exude a sticky glycoprotein called glomalin.
Glomalin, right, which physically binds microscopic soil particles together into aggregates.
Right, which gives the soil its structure, preventing massive erosion during heavy rains. Furthermore, because the fungus is pulling vast amounts of liquid carbon out of the tree to build its own underground architecture, it is a primary driver of long-term carbon sequestration.
So when you look at the forest, the trees are honestly just the vertical solar panels, the actual management of the ecosystem, the structural integrity of the soil, the mineral distribution, the carbon banking that is all being directed by the fungal network.
It absolutely forces us to view the forest not as a collection of individual plants, but as a heavily integrated superorganism, governed entirely by the Wood Wide Web.
The Wood Wide Web. I love that term. And all of this brings us to a really fascinating physiological pivot. Because the intense biochemical warfare required to dissolve solid rock, fight off aggressive underground bacterial competitors, and communicate with the immune system of a giant oak tree. It means that this little mushroom has evolved to be an absolute master chemist.
It's basically a pharmacy in the forest.
Right. It's packed with highly specialized secondary metabolites. And modern medical research is discovering that these specific survival chemicals have profound, potentially therapeutic effects when introduced into the human body.
We are definitely seeing a massive surge of interest in the pharmacological potential of wild fungi. Recent investigations targeting the Terracotta Hedgehog, Hidnum rufescens, and its phylogenetic relatives have mapped an incredibly dense profile of bioactive compounds.
What kind of compounds are we talking about?
We are looking at high concentrations of phenolic acids, flavonoids, tannins, terpenoids, and complex polysaccharides.
Okay, let's isolate a few of these, starting with the phenolics and the whole concept of antioxidant capacity. Because antioxidant is a term thrown around in marketing constantly. You see it on every juice bottle. But in a strict biological context, it's really about neutralizing cellular destruction.
Exactly. Cellular metabolism inherently produces free radicals. These are highly unstable molecules carrying unpaired electrons.
Right.
They aggressively steal electrons from surrounding cellular structures, which damages DNA, proteins, and cell membranes. This is oxidative stress, and it is a fundamental driver of chronic inflammation, cardiovascular disease, and the biological aging process itself.
So to combat this, the body relies on antioxidants, which act as electron donors. They neutralize the free radicals without becoming unstable themselves.
Yes, and the lab analyses of methanolic extracts of Hydnum rufescens show massive radical scavenging potential. They measured the phenolic concentration at over 32 milligrams of catechin equivalent per gram of extract.
32 milligrams. And catechin is the primary antioxidant compound famous in green tea, right?
Yes, exactly. So the density of these protective molecules in the hedgehog mushroom is remarkably high.
And operating right alongside those phenolics are the beta-glucans. These are those heavy, complex polysaccharides found in the fungal cell walls. They don't just pass through our digestive system as fiber, do they?
No, they are actively recognized by receptor proteins on the surface of our immune cells. They modulate the immune response, essentially priming our macrophages and natural killer cells.
Wow.
Furthermore, they act as potent prebiotics, selectively feeding the beneficial bacterial strains in the human gut microbiome, which, as we know, dictates systemic immune resilience.
The immune modulation and the antioxidant density are seriously impressive. But there is a specific avenue of research regarding this genus that almost borders on science fiction: the neuroprotective data.
Yes, this is the big one.
The connection between the hedgehog mushroom and Alzheimer's disease. Let's detail the biochemistry of this for the listener, specifically focusing on the anticholinesterase properties.
Right. To understand the significance of this, we really have to look at the synaptic cleft. That's the microscopic gap between neurons in the human brain.
Okay.
For a memory to be formed or a cognitive signal to be processed, a neurotransmitter called acetylcholine is released from one neuron, travels across the gap, and binds to the receptor of the next neuron.
It's the chemical messenger of learning and memory.
Exactly. But once the signal is delivered, the synapse must be cleared immediately so it can reset for the next signal. So the brain deploys enzymes, primarily acetylcholinesterase and butyrylcholinesterase, to rapidly break down and clear away the acetylcholine.
So in a healthy brain, this is a perfectly balanced cycle of signaling and clearing.
Yes. But in a brain suffering from neurodegenerative conditions like Alzheimer's, the basal forebrain stops producing adequate levels of acetylcholine. The supply of the messenger chemical basically plummets.
And tragically, the cholinesterase enzymes just continue their job, rushing in and destroying what little acetylcholine remains.
Right. The synaptic communication breaks down entirely, leading to the devastating cognitive decline, confusion, and memory loss that characterizes the disease.
It's horrific. So how does the hedgehog mushroom intersect with this neurological tragedy?
Well, researchers tested methanolic extracts of Hydnum rufescens against these specific enzymes, and the results demonstrated that the fungal extract inhibited the action of butyrylcholinesterase by a staggering 63.10% at a concentration of 10 milligrams per milliliter.
Wait, 63%? It actively suppresses the enzyme responsible for destroying the memory chemical.
Yes. The bioactive metabolites in the mushroom bind to the enzyme, preventing it from breaking down the acetylcholine. This allows the neurotransmitter to remain in the synaptic cleft longer, maintaining those vital neural communication pathways.
That is wild.
What is astonishing is that this is the exact pharmacological mechanism of action used by current frontline prescription Alzheimer's medications, like donepezil or galantamine. They are synthetic cholinesterase inhibitors. But here, we have a wild organism synthesizing compounds that perform the exact same highly specific enzymatic blockade.
Okay, I have to step in here with a massive dose of practical reality for the listener, because it is incredibly easy to leap from a biochemical assay in a lab to assuming we have a dietary cure-all.
Yes, we have to be very careful here.
If you go into the woods, forage a basket of Hydnum rufescens, saute them in butter, and eat them, you are not suddenly bulletproofing your brain against neurodegeneration. There is a vast biological canyon between a concentrated methanolic extract suppressing an enzyme in a petri dish and human digestion of a cooked mushroom.
That is a crucial distinction, and it represents the absolute bleeding edge of ethnopharmacological research right now. The 63.10% inhibition was achieved in vitro, using methanol to strip and concentrate the specific bioactive molecules away from the fungal matrix, and then applying them directly to the isolated enzyme.
Right, but when you consume the whole fruiting body, you're throwing all those compounds into stomach acid.
Exactly. Stomach acid, digestive enzymes, and the extreme metabolic filtering of the liver, the first-pass metabolism.
So the bioavailability is the huge unknown variable here. Like, how many of those anticholinesterase molecules actually survive the liver, enter the bloodstream intact, and manage to cross the highly restrictive blood-brain barrier to actually reach the synaptic cleft?
Currently, the in vivo bioavailability of these specific hidden metabolites in humans is just not fully quantified. No responsible scientist is claiming that eating hedgehog mushrooms cures Alzheimer's.
Exactly.
However, the presence of these potent neuroprotective and radical scavenging biochemicals definitely elevates the mushroom from a simple culinary ingredient to a subject of profound medical interest. Consistent dietary incorporation of complex functional foods rich in these specific phenolic and enzymatic inhibitors is widely recognized as a foundational pillar of long-term neuroprotective health.
It is a chemical fortress, basically. And the beautiful irony of nature's economy is that the very chemicals making it a target for medical research are the exact same chemicals that make it so highly prized in the culinary world.
The biochemical overlap is really fascinating. The terpenes and phenolics that the mushroom synthesizes to survive the aggressive soil biome, the exact chemicals demonstrating radical scavenging in human cells, are also incredibly potent deterrents against insect predation.
Which brings us to the actual foraging and gastronomy part of this. Anyone who has ever foraged wild mushrooms knows the absolute heartbreak of finding a massive, beautiful porcini, slicing through the stem, and realizing the interior is just a sprawling apartment complex for fungus gnat larvae.
Ugh, yes, it's completely riddled with tunnels.
But because of its chemical defenses, the hedgehog mushroom is virtually immune to insect infestation. You slice open a mature specimen, and the brilliant white flesh is just pristine.
And that pristine quality instantly makes it a favorite in commercial and high-end kitchens. Gastronomically, it leverages its unique anatomy perfectly. Because of that monomitic hyphal structure we detailed earlier, the total lack of fibrous skeletal tissue, the flesh is remarkably dense and brittle.
Right, it snaps.
Yes, and because of that, it does not turn mucilaginous or mushy when exposed to heat. It holds its structural integrity beautifully during prolonged cooking methods.
So it provides a substantial meaty bite in complex stews or slow-cooked risottos or heavy cream-based fricassees.
Exactly. And the flavor profile is incredibly distinct. It possesses a deep, nutty, earthy sweetness, often finishing with a subtle, almost peppery or faintly oyster-like umami note.
But foragers really have to navigate the timing of the harvest carefully, don't they?
They do. While mature specimens are large and structurally sound, they develop an accumulation of secondary metabolites as they age. And that translates to an intense bitterness on the palate.
The older they get, the more chemical armor they build up, essentially making them bitter.
Right. So the classic culinary trick is to take a paring knife and gently scrape the spinos hymenophore, the stalactites, completely off the cap of older specimens before cooking, because the highest concentration of those bitter compounds resides in the spore-producing tissues.
It's an essential prep step if you've got a large harvest. But really, the ideal culinary targets are the young, newly emerged mushrooms, right? The ones often called buttons, where the flesh is sweetest and the bitterness hasn't developed yet.
Yes, the buttons are highly sought after. But targeting those young buttons introduces the darkest, honestly most terrifying aspect of foraging this genus.
Yeah. Let's talk about the danger of lookalikes. We established that the defining morphological feature of the hedgehog is its teeth. But when the mushroom first pushes through the soil in its button stage, the cap is tightly curled inward.
And the spines on the underside have not fully developed. They can look like a totally smooth surface or even resemble rudimentary gills.
Which is terrifying, because this morphological ambiguity in the juvenile stage is a massive safety hazard. A novice forager looking for young, sweet hedgehogs can easily confuse these spineless buttons with several highly toxic species that share the exact same terrestrial habitat and that same terracotta orange cap coloration.
And the most devastating of these lookalikes belong to the genus Culinary, specifically the species collectively known as the deadly webcaps.
The deadly webcaps, I mean, the name alone is a massive red flag. But the biological mechanism of their toxicity is truly insidious. It's not just a stomachache.
No, it's horrific. The Culinary species, synthesize a potent nephrotoxin called orellinine. If a forager misidentifies a young webcap button as a hedgehog and consumes it, the true horror of orelline poisoning is its extreme delayed onset.
Delayed onset, meaning you eat it and nothing happens immediately.
Exactly. It does not cause immediate gastrointestinal distress. The toxin is quietly absorbed and specifically targets the epithelial cells of the proximal tubules in the kidneys. It interrupts RNA synthesis and generates massive amounts of localized reactive oxygen species, silently tearing the renal cells apart at the molecular level.
And the victim has absolutely no idea this is happening inside their body.
None. The latent period can last anywhere from 3 days to over 3 weeks.
3 weeks?
Yes. During those weeks, the individual feels perfectly healthy while their kidneys are undergoing irreversible necrosis. By the time the physical symptoms actually manifest, which present as an unquenchable burning thirst, severe nausea, and intense pain in the lower back room, the renal destruction is already catastrophic.
The kidneys simply shut down.
It results in delayed acute renal failure. Treatment often requires lifelong hemodialysis or a complete kidney transplant, and in many historical cases, it is entirely fatal.
It is a terrifying biological trap. You eat a mushroom, feel totally fine for 20 days, and then your organs just fail. This is exactly why the absolute, unbreakable, non-negotiable rule of foraging this genus is that if the specimen does not have clearly defined, distinct spines hanging from the cap, you do not pick it.
Period. You never assume a smooth button is a hedgehog just because it's growing near other hedgehogs.
The spines are the singular guarantee of safety.
They absolutely are.
Okay, so given the extreme hazards of wild foraging, combined with the massive culinary demand and the exploding medical interest in its biochemical properties we talked about, the logical conclusion for modern agriculture would be to industrialize it, right?
I think so, yes.
I mean, we factory farm white button mushrooms, shiitakes, and oysters in massive climate-controlled warehouses on sterilized blocks of sawdust and grain. So why hasn't the agricultural sector industrialized the hedgehog?
Because we cannot industrialize the wood wide web. This brings us full circle back to the biological chain of the obligate ectomycorrhizal relationship.
Right, the need for a living tree.
Exactly. You cannot trick a hydnum spore into digesting sterilized sawdust in a dark room. It simply lacks the enzymes. It absolutely requires the highly specific, complex carbon exudates provided by the roots of a living, photosynthesizing forest giant. Furthermore, it requires the complex, competitive microbiology of the natural soil biome just to trigger its fruiting cycle.
You really can't separate the architect from its environment. Factory farming is biologically impossible.
It is. However, the commercial demand is driving a very sophisticated frontier called forest farming. This is a deeply holistic agricultural approach, closely mirroring the multi-generational, highly guarded techniques used to cultivate truffles in the Mediterranean.
So you don't farm the mushroom, you curate the forest.
Precisely. You manage the entire woodland ecosystem to heavily favor the natural propagation of the local hydnum lineage. This involves profound ecological engineering on the human side.
What does that look like in practice?
Foresters selectively thin the tree canopy to dial in the exact light levels reaching the forest floor. They manage the understory brush to regulate soil moisture and temperature fluctuations. They carefully monitor the soil pH. In advanced operations, they will actually take young, native saplings, heavily inoculate their root systems with the spores of the specific, locally adapted hydnum species, and plant them back into the managed plot.
Essentially seeding the subterranean economy.
Exactly. It requires an absolute mastery of the local genetics and the local geology we discussed today. You have to nurture the host tree, protect the glomalin structures in the soil, and ensure the fungus has the exact mineral profile it evolved to dissolve.
It is the ultimate expression of working within nature's parameters rather than trying to force nature into an industrial box.
It really is.
Which provides the perfect synthesis for the incredible biological journey we've mapped for you today. We began by looking at a supposedly simple, weirdly shaped mushroom with a sweet tooth, assuming we basically understood its place in the world.
And instead, we uncovered a genetically complex, ancient ecological engineer. We saw how multi-locus DNA sequencing totally shattered the holarctic myth of a single global species, revealing a hidden map of dozens of highly localized endemic lineages.
We excavated the mechanics of the wood wide web, detailing how these localized fungi act as deep earth miners, utilizing powerful organic acids to dissolve solid rock. and trading the extracted minerals across the Arctic net in an encrypted chemical exchange for tree carbon.
We analyzed their potent chemical fortress, discovering incredibly dense antioxidant capacities and anticholinesterase properties that actually mirror synthetic Alzheimer's medications while also providing absolute immunity against insect predation.
And we translated that biochemistry into the culinary world, exploring its unique monomitic texture in the kitchen, while starkly warning against the fatal, delayed renal destruction caused by confusing young buttons with the deadly Cortinarius webcaps. It is an organism that perfectly encapsulates the staggering, invisible complexity of the natural world.
It is a true masterclass in localized evolutionary adaptation.
But I want to leave you, the listener, with a final, provocative thought regarding that extreme specialization. We've established that these hidden species are the ultimate biological specialists. They have spent tens of millions of years perfectly, stubbornly adapting their biochemical toolkits to dissolve the exact mineral profile of their local soil while fine-tuning their chemical communication to match the roots of specific local trees. They are deeply biologically anchored to a localized reality.
Very deeply anchored.
But we are currently living in an era of unprecedented rapid climate change. The temperature bands and precipitation zones of the planet are shifting poleward at a velocity that far outpaces the natural migratory speed of ancient forests.
Which presents a massive problem.
Right. What happens to the ultimate specialist when its specific world simply disappears? If the host trees are forced to migrate to survive, but the soil chemistry in the new location doesn't match the fungal toolkit, or if the climate changes faster than the neutral mutations in the fungal DNA can adapt, will these ancient subterranean ecological engineers be able to rewrite their chemical blueprints in time? Or will the invisible infrastructure holding the forest together quietly collapse beneath our feet?
It's a sobering question.
The next time you walk into the woods, look past the skyline of the canopy, look down at the soil, think about the massive, ancient engines humming silently beneath your boots, and ask yourself just how fragile that deeply specialized machinery really is. Thank you for joining us on this deep dive. Stay endlessly curious, always check for the spines, and we'll see you back underground next time.